So हमारा आज का टॉपिक affinity chromatography, and first of all we will discuss its introduction. Affinity chromatography is a unique and powerful technique which is used for purification of different biomolecules on the basis of their interactions with some immobilized ligands in the stationary phase. So what happens here that we bind some ligand with the stationary phase, and when some mobile phase passes through it, some components present in the mobile phase, they get attached, they interact with these immobilized ligands present in the stationary phase, and in this way those compounds, they are bound with these ligands. Now when all of the rest of the components, they pass through the column, what happens? Then we change the conditions of the column, and these bound biomolecules or compounds, they get separated from the ligands present in the stationary phase, and then they pass out, they elute, and they can be collected. So it is a technique for purification of biomolecules on the basis of their interactions with these ligands present in the stationary phase. Now, interactions of these biomolecules with the ligands, they are similar to like some enzyme is bound with substrate or some antibody is bound with some antigen. And this technique is used to isolate pure substances present at low concentration in some large volume of crude sample, or it is also used to remove the contaminants present in the sample. This technique is also used to separate active biomolecules from the denatured or functionally different forms. So there may be different forms of the same compound, and some of the forms may be denatured, or they may be functionally different from the functional form. So what happens with this technique? We use to separate the denatured form from the normal or from the functional form of the compound. So it is also used to separate functionally denatured form of the compound from the normal form of the compound. And proteins, they are separated on the basis of reversible interaction between a protein and specific ligand coupled to chromatographic matrix. I have already mentioned that when rest of the components, they pass through the column, the bound compound with the ligand that is separated, or it is separated, the conditions, they are changed. For example, pH, for example, ionic strength, they are changed, and in this way the bound ligand that gets separated from the stationary phase, and then it passes out from the column and it can be collected. So rest of the components, they pass first, and the bound compound, that is eluted at later, which can be collected. This method offers high selectivity and high resolution for a protein of the interest. Of course, from the mixture of a compound in which there may be different components present, what we do, we separate a particular compound, for example, a particular protein, with high selectivity and with high resolution. And this purification and recovery of active material is generally very high if we compare this technique with other techniques.